I'm Masaki Mizutani from the AIST Japan. Today I will introduce about the laboratory, adaptive laboratory evolution of minimal cell to room temperature. The Syn3B has been used for functional analysis by reconstitution of introduced gene function. For example, the spiroplasma swimming reconstitution or urea plasma adhesion reconstitution. This strategy is really cool for us. And to precisely reconstitute the function, we should precisely express and fold proteins. Here we think in case that reconstitute the mycoplasma mobile grading. At first, we choose the grading related to genes and transform into Syn3B. And if it's successful, then we can reconstitute grading motility in Syn3B. But here we have one problem. The problem is the differences of optimal growth temperature. Because the optimal growth temperature of mobile is around 25, but the optimal growth temperature of Syn3B is 37. So also the optimal growth temperature of the disk transformer is also 37. And these differences of temperature causes sometimes the protein misfolding. It's the resulting failed to reconstitution. So not only mycoplasma mobile, in the many environmental molecular species have the lower optimal growth temperature than 37. For example, the spiroplasma in insect or phytoplasmas. So question here is, can Syn3B grow at lower temperature? So at first, we checked the growth speed of the original Syn3B strain at various temperatures. This is the kind of the growth curve, and this is the doubling time in exponential phase. And Syn3B has the fastest growth speed, 37, and the growth speed decreased with temperature. And finally, at 27 degrees, the strain has shown no significant growth. So it means that also at the 25, they can't grow. To make the lower temperature culturable Syn3B, we do the passage cultivation. At first, I inoculate the original Syn3B and cultivate at 30 degrees because it can slowly, but it can cultivate at 30 degrees, this green one. And after the cell growth, we picked up the cultivated cell and inoculated into new sp4 media and cultivate at 30 degrees. And we continue this cycle 20 times. This is the graph. We do individually 4 lineages. It means the individual 4 tubes. This experiment, I show the average here. And this axis is the cultivation time until passage. It's a kind of the growth speed. Until the prime passages, the cell can grow for around 100 hours. And when we passaged, the growth speed become faster. But finally, the 20 passage strain have around twice faster growth speed than the original cell. And next, we try to cultivate the 20 passage strain at the 25 degrees. And surprisingly, it can grow at 25 degrees. So we do this passage cycle also at the 25 degrees. And in 25 degrees, it cell have the faster growth speed when we passage the cell. The next week, compared the growth speeds in the 3 strains. The one is optimized at 37, the one is optimized 30, the one is optimized 25. And we cultivated these 3 strains at the 37 degrees. The 37 optimized one have the fastest growth speed, and the 30 degree optimized one have a little bit slower, and the 25 degree optimized one have the slowest growth speed. And we do the similar experiment at also 25 degrees. At 25, the 37 optimized one show the never the grow, and 30-degree optimized one have the a little bit slow but grow, and 25 optimized one looks the nicely the grow. So it means the optimal growth temperature of adaptive strain shifted to the lower the temperature the ranges. And to understand the mechanism of low temperature optimization, we performed proteomics analysis for original Syn3B at low temperature optimized strain and compare the expression level of each protein. And we do the passage individually for lineage. So we show the 4 lines, vertical lines, and this is the CDS, the name of the annotation. And if the adaptive strain have higher expression level compared to original one, that protein is showed red. And when the adaptive strain have the lower expression level than compared to the original strain, it's shown blue, something like that. And to better understand, we categorize these the CDS into the groups, the low or the pathways. For example, the glycerol metabolism increase and the RNS increase, something like that. And also the transporter or the lipoprotein increase, or something like that. And the chaperone and the peroxidase, the decrease, something like that. And from here, we introduced the 3 important mechanisms for the low temperature optimization in our adaptive strains. The one is the RNS, because this is the well-known the mechanism of cold shock response in E. coli. When we put the cell at low temperature, first the messenger RNA folds the abnormal secondary structure, and it causes the failed to translation. So the cell should actively degrade the misfold the messenger RNA and transfer to the nucleotides the recycling pathway. So at that time, the E. coli the highly expressed RNAs. And here I also show the name of RNase for RNA degradation in the Bacillus. And here is our results, the adaptive strain of Syn3B. They have the higher expression level that these RNAs, it does similar to this E. coli situation. So it means the Syn3B has the similar adaptive system for low temperature like E. coli. And next is the related to DNA replication. Because at the low temperature, annealing of double-strand DNA is excessively stabilized. So it causes the stuck of the DNA polymerase. So we should resolve this annealing. And this is the result of the adaptive Syn3B. They have higher expression level of the DNA helicase B and also single-strand binding proteins. So to express higher these proteins, the Syn3B clear this strong annealing, and it's helpful to the effective DNA replication. This is very important for the low temperature adaptation. But this is hard. This is here I show the glycerol metabolism. The left panel shows the lipid metabolism pathway in Syn3A, and this red enzyme have the higher expression level in the adaptive strain. So it means that the pathway may be the increase. That this pathway is the transform glycerol to glucose 6-phosphate. Maybe it's the help to accelerate glycolysis, and it's helpful to produce more ATP molecule to help cell growth at low temperature. This is the last slide, the conclusion of this study. Now we have the 37 Syn3B and also 30-degree adapted one and 25-degree adapted one. So now we can choose a suitable strain for individual donor bacterium. And that's not shown, but this is the flow temperature adapted strain can transformable. We checked already. And for future industrial use, this 25 adapted syn3B is very beneficial because we can save the cost of the heating incubator. Yeah, that's it. Thank you for your attention.
Masaki, so there were recent papers. I was an author on both. One from the University of Indiana, where they did adaptive evolution of syn3B, and then from UCSD, where they did adaptive evolution of syn3A. And they showed that, so if you think about it, the organism on which the minimal cell was based, Mycoplasma mycoides, it had never been adapted for growth in SP4 media. People wanted to keep it as close to the pathogen as possible, so it was always minimal passage in the past. And so now these groups at Indiana and UCSD have adapted both the wild type organism and the minimal organism for growth in SP4. Are any of the changes that you see from in your adaptation to your growing the cell in SP4, are they the same changes that people see at the 37-degree amplification? So extended growth of the cell at 37 degrees produced a number of changes. Are these consistent with any things that you saw?
Yeah, thank you for the question. Our adaptive strain also have the differences expression in the FTSZ. Maybe this paper mentioned about the many FTSZ changes. So it is the similar point. I think. And also the mutation in ABC transporter, they mentioned, and we can see also the mutation in ABC transporter. It's also the similar things. So that we can see the several similar things.
Okay. Other questions for Masaki? Are you going to sequence these genomes?
Yes, we did.
Okay.
So I guess
those proteomics changes.
Yeah, the mutation in genome is the less than the proteomics changes, but several strains have the mutation in the RNA polymerase and also sigma factor. Maybe it's one of the point of this adaptation.
Okay. How fast and can you remind me at 30 and at 25 degrees, what were the growth rates, the doubling times?
This right?
So but what are I'm curious what the doubling times are.
We didn't check the critical number, but normally the 1% the inoculation, the normal original syn3B, become full growth at 1 to 2 days or something. And our 25 optimized one, the cultivate at 25 degree, to be the fully growth, it takes 7 or 8 days.
How long does Mycoplasma mobile take to double?
Around 7 hours.
Okay. We're not in that league.